Hi everyone, I'm now going to go through the MCQ, first 10 questions of 2019 paper. Let's begin with question 1. Question 1, they ask which statements about triglycerides and cholesterol are correct. Look at statement 1. Fatty acids in a triglyceride may be saturated or unsaturated, but in a cholesterol, they are always saturated. Okay, the error in this statement is in that saying that it's always saturated in cholesterol. If we were to look at the diagram of a cholesterol model, you often find that there are a lot of rings. And if you look carefully into the rings, you go and refer to one of the models, you'll find that they often come with double bonds. These double bonds mean that they are actually unsaturated sometimes. Okay, let's look at statement 2. Triglycerides and cholesterol both have a hydrophobic region. Is this true? This is true. And that's what also makes lipids. Lipids have a lot of hydrophobic regions. But not necessarily that they do not have any hydrophilic regions. Because we know, for example, phospholipids as well as cholesterol, both of them are amphipathic. They have both hydrophobic and hydrophilic regions. So last statement, triglycerides are non-polar molecules, cholesterol are polar molecules. And I just said earlier that cholesterol, actually, if you look at it, often it comes with a OH. This OH makes it, this part, polar. But actually, a lot of it is non-polar. Therefore, it makes it amphipathic. So it's not right to say that the entire molecule is polar. Okay, so the best answer is A. Let's move on to question 2, which describes the chemical reaction when sucrose is digested. Okay, when we say that something is being digested, we mean to say that we are breaking it down. So the best process for this is hydrolysis. Hydrolysis. Okay, not condensation. So I'll first need to rule out A and B. So let's look at hydrolysis. What is hydrolysis? In hydrolysis, you will actually use water. The word hydro means water. That means water is being used. Lysis means to break. So you're actually using water to break the bond. And therefore it cannot be C, because C says that water is produced. So the best answer is D. Let's go to question 3. Which statement describes how the structure of starch is suited for its function? What is the function of starch? The function of starch is for storage. Storage of what? Storage of glucose, because many glucose molecules can condense together to form starch, which is a polysaccharide. There are two forms of starch, amylose and amylopectin. So let's have a look at the statements. A. Amylose and amylopectin are insoluble and do not affect the osmotic properties of the cell. Is this true? This is true. Alright, we know that glucose in itself, they come with actually a lot of OH groups, right? This makes them actually very soluble in water because all these OH groups will like to interact with water, very hydrophilic. But the interesting thing about when all these glucose condense together to form a polysaccharide, so I'll draw a very simplified model. Okay, imagine all these glucose coming together to form a polysaccharide, okay, many more glucose. When they start to condense together, they will actually condense together. Okay, I'm going to now zoom in. They'll condense together to form a helix. Okay, it adopts a very helical conformation. And what's interesting is that all the OH groups will end up pointing inwards. So if I were to zoom in a bit more, the OH groups will be pointing inwards. Okay. So even though OH groups is polar and will be attracted to water, hydrophilic, because they're all pointing inwards, actually any water molecule that is nearby won't really get to interact with the OH groups. So that's what makes it not quite able to interact with water. On another note, we also say that it's insoluble because other than it not being able to interact with water, amylose and amylopectin, to dissolve something, you need the individual glucose molecules to be free to be able to dissolve. The glucose is now embedded in this polysaccharide. Such a large molecule cannot dissolve. Okay, so if it cannot dissolve, it will not affect the osmotic properties. Osmotic properties is referring to the water potentials. when something is not dissolved, it shouldn't affect the water potential. Okay, so the best answer is A. But let's look at the other options, B, C, and D. Amylose has a branch structure, and amylopectin is coiled to give a compact molecule for transport. Okay, what's wrong with this statement? Amylose is not branched. Okay, in fact, it's only helical, but there aren't any branches coming out. Amylopectin, while it is coiled, it is not for the purpose of transport. Okay, what is transported in plants is sucrose. Starch, such as amylopectin or amylose, is too big, very hard for transport, better for storage. So B is out. What about C? In the breakdown of amylose, many hydrolysis reactions allow release of energy in the form of ATP. Now, when we break down amylose, yes, it's hydrolysis, but we do not release energy in the form of ATP when we break down starch. We release energy more in the process of cellular respiration, right? But we do not do so when we break down starch. So C is out. D also, on a similar note, is very odd to say that many condensation reactions to form amylopectin will release energy. There's no such process. Okay, so this is also wrong. The best answer is A. Let's now look at 4. Which substance contains carbon, hydrogen, oxygen, and nitrogen? Actually, rule of thumb is that a lot of biomolecules come with carbon, hydrogen, and oxygen. Some biomolecules come with nitrogen, and examples of that are nucleic acids. The nucleic acids are made up of nucleotides. Nucleotides also contain nitrogen in their nitrogenous bases. Okay, you may have heard this before, nitrogenous bases. Examples of nitrogenous base is A, T, C, G, and U. These are all nitrogenous bases, right? All nucleotides come with a sugar attached to a base and a phosphate group. Okay, and this base has a lot of nitrogen. Last but not least, proteins. Proteins also can come with nitrogen, right? We know that there's an amino group. So out of 1, 2, and 3, which one will have nitrogen? ATP. Actually, ATP is a form of nucleotide. Okay, the T just means that there are 3 phosphate groups attached. Okay, 2, deoxyribonucleic acid. This one, DNA, it comes with nitrogen, yes, in all the bases. Last but not least, 3, hemoglobin. Hemoglobin is a protein, so proteins will have nitrogen. And the best answer is D. Now let's look at question 5. The graph shows the effect of pH on the structure of a protein, which consists of 1 polypeptide chain. And you see the word alpha helix? It is saying that between pH 0 to pH around pH 6, this alpha helix, which is a secondary structure, is present in the protein. Beyond pH 6, you notice that this secondary structure becomes a random coil. No longer this very structured alpha helix. So which statement is true? At pH 2, the protein has lost its secondary structure. At pH 2, actually the secondary structure, this alpha helix, is still present. So A is wrong. At pH 2, the protein has lost its tertiary structure. We can't tell from this particular graph. This particular graph only gives us enough information to know about what happens to the secondary structure, which is this alpha helix. So we cannot tell. C. At pH 10, the protein has lost its primary structure. At pH 10, says that now it's a random coil. But I did not say that the polypeptide has broken down into its amino acids. This random coil still means that there's a primary structure, just that it's not coiled up properly. So C is also not right. At pH 10, the protein has lost its secondary structure. This is true. From an alpha helix, which is the secondary structure, to now just the primary structure. So the secondary structure is gone. Best answer is D. Now let's look at question 6. Question 6, which correctly describes a DNA molecule? A. It contains 2 strands of orientated parallel to each other in a 5 to 3 direction. The word parallel is the one that's wrong. It's supposed to be anti-parallel. B, the amount of thymine and adenine will be approximately equal. This is true because if we look at a complementary strand, for every A, you should find a T. For every C, you should find a G. So overall, you should find roughly the same amount of adenine and thymine. We say approximately equal because when we actually conduct experiments to try to determine the percentage of A and T, usually you will not get exactly the same. This is because of human error when we are trying to determine it using experiments. C, the nitrogenous bases bind together via phosphodiester bonds. This is not true. Okay, what is the nitrogenous base? Again, if we look at a nucleotide comes with bases, a sugar, as well as a phosphate group. If I were to link many of these nitrogenous bases together, you will get a strand of DNA. And each strand of DNA, right, if this is the base can be A, T, C, or G, right? What you see is that it can be A, T, C, maybe a G, then maybe another G, so on and so forth, right? So to say that, okay, let me just write the rest down. To say that the nitrogenous bases bind together via phosphodiester bond, okay, the nitrogenous bases between A and T, they do not interact via phosphodiester bonds. They interact via hydrogen bonds. So what do phosphodiester bonds link up? They link up this sugar phosphate backbone. So this is what links nucleotide to nucleotide. That is the phosphodiester bond. If I were to zoom in, you will find that this will be bonded to a phosphate group and then to another sugar group, and then there's another base. Okay, maybe this one is T, as seen over here. Then will be linked to another one, right? So this bond over here is a phosphodiester bond. So C is out. Last one, the sugar phosphate backbone consists of nitrogenous base, phosphate group, and a ribose sugar. The error in this statement is in the ribose sugar. In DNA, rather than a ribose sugar, you get a deoxyribose sugar. Deoxy means that it does not come with an OH group. Okay, in the case of RNA, you find that there will be one more OH group at carbon number 2 of the sugar group. Okay, so this option is also not right. Best is B. Let's go to to question 7. In question 7, they show you a series of results from a series of food tests. Then they ask you which nutrient does this food contain. Purple color in the Bure's test indicates that protein is present. So anything with protein could be an answer. Purple color when heated with Benedict solution. This result is a little odd. We can't quite tell what is present when we see purple color in Benedict solution. After Benedict solution, you tend to see results like blue, which means reducing sugar is not present, or you may see a brick red or orange, which means reducing sugar is present. So this one is unknown. Yellow color with iodine means that starch is absent. And white emulsion in ethanol means lipid is present. So fat must be present. Fat must be present. Right, so the best answer is A. Let's look at question 8. Question 8 shows you a graph. A typical graph showing the energy required for a reaction to continue. This is the amount of energy that the reactants have. And this is the amount of energy that the products have, okay, at the end of the reaction. Very often we will need a certain amount of energy to be overcome before the reaction can proceed. And we call this energy the activation energy. So when there's an enzyme present, what does the enzyme do? The enzyme helps to lower the activation energy. Alright, less energy is needed to be overcome. Okay, so what the enzyme does is it helps to lower number 3. Okay, reduces the amount of energy required. Now it's less when the enzyme is present. So best answer is C. Let's look at question 9. Question 9 shows you a scenario, a case study. It says that in farming practice, freshly picked corns are often immersed in boiling water. Okay, in the moment you see boiling water, you want to think of temperature. Temperature is high. This can help preserve the sweetness of corns because sugars in the corns, the sugars, will not be converted into starch. That means some sort of a reaction needs to occur, typically. Typically, what they're implying is that the sugar will undergo some sort of a condensation reaction to form starch. Okay, so this condensation reaction doesn't seem to occur at high temperatures. Why is this so? I immediately think of enzymes, right, when temperature is involved, and then when reactions do not proceed. Because enzymes, at the end of the day, they are proteins, and proteins are affected by high temperatures. They become non-functional. So D is the answer. This protein likely helped to condense the sugars to form starch. But what are the other options? The boiling changed the molecular structure of the sugars. Okay, I'm not quite sure how changing the molecular structure of the sugars will affect its conversion to starch. It's unclear. B, the boiling denatured the protein channels in the cell membrane. Okay, this has nothing to do with the conversion of sugars to starch. Okay, even if you affect the protein channels do not convert the sugars to starch. C, the boiling destroyed the vesicles for storing starch grains. Okay, also not quite related. How does the vesicle affect the conversion of sugars to starch? So D is the best answer. Anyway, vesicles are made of lipids. So the temperature shouldn't, destroy the lipids, unlike in proteins. Last, let's go to question 10. When an enzyme is formed from amino acids and is released out of the cell, it has to pass through some organelles or membranes. Which shows the correct sequence of organelles or membranes that have been passed through. Okay, so the sequence will go like that. Ribosomes is involved in translation, makes use of amino acids to form polypeptides. These polypeptides can then be deposited inside the endoplasmic reticulum, likely the rough endoplasmic reticulum, where the ribosomes are attached. Then the vesicles are bud off as transport vesicles towards the Golgi apparatus. From the Golgi apparatus is packaged and modified, and after that transported buds off as secretory vesicles to the cell membrane for release of what we call that process of release secretion. Okay, so the best answer is C. You should start off with ribosomes because that's, after all, what produces the polypeptide. Okay, and we say enzyme is formed from amino acid. That's still okay because we know enzymes are protein in nature. Okay, so that's